In this video, we are going to use ProteinTools, a comprehensive toolkit for analyzing protein structures in detail. With it, you can explore hydrophobic clusters, hydrogen bond networks, salt bridges and charge segregation, and even generate contact maps to visualize residue interactions. Now, copy the web page URL and paste it into your preferred web browser to access the site. Within ProteinTools, you can perform four main analyses: hydrophobic clusters, hydrogen bond networks, salt bridges and charge segregation, and also generate contact maps to visualize residue interactions. We'll begin with hydrophobic cluster analysis. Simply enter the PDB code of your protein, or upload your predicted structure to get started. The analysis will provide you with the total number of hydrophobic clusters and show the precise locations of these clusters within the protein. You can download the list of hydrophobic clusters in Excel format, or export a PyMol session to continue your analysis in more detail. Remember, these analyses aren't limited to crystal structures. You can also explore your computationally modeled proteins in the same way. Next, let's examine the hydrogen bond network. Similarly, you can either enter the PDB code or upload your own modeled protein structure for analysis. This analysis provides detailed information about the hydrogen bonds, including how many are present, where they form, and which amino acid pairs are connected by each bond. Similarly, you can download the hydrogen bond list or export a PyMol session for further analysis. Now, we'll explore salt bridges and charge segregation. These analyses operate just like the other tools in this browser, making it easy to study your protein structural features. Keep in mind, you can modify the protein structure display to better visualize the features you're analyzing. You can also perform contact map analysis, just like with the other tools in this toolkit. Before we finish, a quick shout out to the institutions and agencies behind this platform. We've also added a scientific citation if you want to explore the topic further. Thank you for watching. If you have any questions or inquiries, drop us a message in the comments below. We'll be happy to help and respond as best as we can. If you found this tutorial helpful, make sure to like, share, and subscribe for more content.